Thank you very much, Masa, for agreeing to give the Buildacell seminar, and you can get started.
Okay. Okay, so I will start my presentation. So I'm Masahiro Takinoue from Tokyo Tech. Thank you so much for inviting me to this wonderful seminar today. So today I'd like to talk about the artificial cell engineering using DNA droplet technology. Okay, so let's move on to the next slide. Okay, so as you know, the living systems are very dynamical. So the left one is one of the famous movie of the neutrophil chasing the bacteria. The neutrophil shows non-equilibrium dynamics such as locomotion and deformation and phagocytosis also. The right one is cell division, as you know. This shows the non-equilibrium dynamics of the reproduction of cell driven by some kind of enzymes and motor proteins. So what we can understand from these examples is that living cells are some kind of the dynamical and soft and fluidic biomolecular assemblies with intelligence. So that you know that even the living systems are constructed with molecules. However, the huge gap exists between the living systems and the non-living matter. So no matter how sophisticated biomolecular nanostructure we see, they obviously cannot compare to living systems such as cells. Therefore, we need a new science to bridge this gap. So that is a construction of the artificial cells, maybe. In a sense, this is also the question of what life is, and I believe we will understand it when we can bridge this gap. So I believe qualitative leap, not mere just the complexity, will be necessary to bridge this gap. So here, I think that kind of the leap would be the information processing and autonomous dynamics of the soft materials. So that we can say living systems are matter in which the information is always autonomously created and processed. That is inseparability of the information and the matter characterizes the living systems. Also, the living systems have the properties of being able to control oneself without needing other controls. Such autonomy can be realized in non-equilibrium systems. As a result, unlike mere matter, those properties can produce living phenomena such as learning, the adaptation, the evolution, or something. So the key point is I think the programming of molecular assemblies to create the dynamics and to create the intelligence and the micrometer scale non-equilibrium conditions. So in bridging the gap, it must be noted that the cells are not merely a diluted solution in a flask. So the living cell is some kind of the condensed viscous micrometer scale droplet of the biological soft matter. Actually, we can find droplet-like structures everywhere in the living cells. For example, this example, the cell cytoplasm and nucleus and many organelles are biomolecular membranous droplet enclosed with lipid membranes. And also this is a chromosome, the nucleus and RNA granules are biomolecular membraneless droplets formed by the coagulation and or the gelation or the liquid phase separation by polymers. So we have to develop the methodology to program such the condensed viscous micrometer scale droplets actually. So the creating artificial cells is just a programming of the non-equilibrium intelligent and dynamic behaviors of the biosoft matter droplets, such as the sensing, the computation, and the locomotion and deformation, something. So the program must not be the rigid, because of the flexibility of the living systems, but must be the inseparable from the soft matter as a body and flexibly changeable in response to the environment. So the to create the intelligent and dynamical behaviors, I have a question. So what is the meaning of a membraneless droplets? Membraneless droplets is just a condensate of the biomolecular polymers or something without the lipid membrane. Okay. Thank you for the question. So the to create intelligent and dynamical behaviors, the chemical to mechanical or chemical to information energy transaction in non-equilibrium are essential. Maybe. So this is the minimal non-equilibrium concept of the artificial cells rehearsed by the many researchers actually. Here, the substrate S that is introduced in a compartment. S is transduced to a product P by some kind of chemical reactions. And also the P is degraded to a waste W by a chemical reaction too. If the addition of the substrate S is sustainable, the sustainable non-equilibrium dynamics is produced. But if S is not sustainable, the only temporary non-equilibrium dynamics will be observed in the system. So from this viewpoint, our group is seeking to the physics on the soft matter droplet to build the artificial cells. It consists of the maybe 5 parts. The first one is the designing and the construction and the understanding and control of the system and the application of the artificial cell system to the actual application, some kind of the engineering, something. Okay. So recently, not only as you know, not only our group, but also many research groups that have been exploring the various dynamical artificial cells that are driven by the chemical or the enzymatic reactions under the non-equilibrium conditions based on the similar concepts. For example, this is the vesicle, the deformation of the active pneumatic vesicles with kinesin microtube motors. This is a cell-sized molecular robot that switching its motion by the light irradiations. This is one of the example of the self-propelled oil droplet that move by consuming the fuels. So more recently, the DNA-based droplets have been used to achieve the dynamical droplets. So this kind of DNA-based droplets are useful in terms of the programmability and the flexibility to develop the dynamical artificial cells. So for example, this is the enzymatic degradation and bubbling of DNA droplets with a spontaneous motion like this. So this is the spontaneous division of DNA droplet by our group. And also this is a locomotion of the DNA droplet with some kind of the biochemical reaction networks. Okay. So at the first time, the DNA droplets and the DNA hydrogels have been explored as new materials and new phases in DNA nanotechnology. That means relatively the static, the equilibrium features. The research on the constructing the dynamical the behavior is still open problem and ongoing project actually. So in my talk, I will focus on such a dynamical DNA droplet research for non-equilibrium artificial cell the construction. So these are the today's topic maybe. So let's start the first topic, the dynamical behavior of the DNA droplets. So previously, my former postdoc, Dr. Sato, demonstrated that this kind of the Y-shaped DNA motif assembled into a phase-separated droplet of the DNA. So this Y motif, Y-shaped DNA motifs are connected through the sticky end here, and the Y motif can self-assemble into this kind of the DNA-based hydrogel or DNA droplet. As you see, in the state of the DNA droplet, they can fuse with each other like this. So this is stained with fluorescent molecules. So this is a DNA-based droplets like this. This is very fluidic, and if it collides, they can fuse with each other like this. It's very fluidic. Okay, so and we next design the stability of the sticky end part by changing the length of the sticky end without changing the other part. So here the changeable part. So if we elongate the sticky end part, the stability increases like that. And also we make this kind of the phase diagram of the DNA droplet and the DNA gels. So if the stability increases, the transition point between the droplet and the gel state is also increases like this, and also the dispersion and the droplet phase also, and that point is also increases like this. In addition to this kind of the feature of the DNA droplet and the DNA hydrogel, we investigated some kind of dynamical parameters such as the surface tension of the DNA droplet and the viscosity of the DNA droplet at some kind of conditions. So we can control such many properties based on the DNA the sequence design. So the first example of the interesting designing is the orthogonal DNA droplet formation. So we can control the fusion of the two DNA droplets based on the sequence at the sticky end part. So if we made the non-complementary sticky end part here, they cannot hybridize with each other. So the two DNA droplets cannot fuse with each other. So this is the result. So the green droplet and the green droplet can fuse with each other, and the blue one and the blue one can fuse with each other like this. But the blue one and the green one cannot fuse each other like this. So that we can control the direction of the reaction, and then control the fusion properties based on the sticky end sequences. So as we saw, these two droplets cannot fuse directly, but when we add the cross-linking DNA motif composed of the 6-branch linker motif, these droplets can fuse with each other through this 6-branch linker. This is the result. You can see the DNA droplet fuse with each other. This includes 3 types of the DNA motifs inside. Okay, so we can control the fusion ability and orthogonality based on the DNA stick end or the motifs compositions. So then the next we design the spontaneous division of the DNA droplet by changing the 6-branch linker a bit. Here only the center part of the 6-branch linker was changed to the RNA regions, RNA sequence. So when RNase, this is the RNA degradation enzyme that is introduced into the solution, this 6-branch linker is cleaved into 2 portions of the 6-branch DNA, and this linker loses the cross-linking ability. So the DNA droplets shows the spontaneous phase separation, and finally it will divide into 2 droplets. So this is the result. So as you see, the DNA droplets divided into the two parts of the droplet, including only green or only the blue parts like this. So we can control this kind of the self-division properties of the DNA based on the motif design. So that we can reconsider this phenomena in the energetical view. So I'd like to consider the role of the 6-branch linker as a fuel substrate as well as linker. So at the initial state, by the binding free energy of the linker substrate S, the DNA droplet exhibit a mixed state like this. This is the higher energy state. But the linker cannot spontaneously react so fast because of the high barrier here. But after the enzyme addition, the reaction starts and produce a cleaved product like this. So the reaction starts and then it cleaves the inside of the droplets. Simultaneously, the phase separation of the 2 orthogonal DNA to orthogonal DNAs starts, and finally the DNA droplet divided into 2 types of the DNA droplet like this. So this means that by releasing the linking binding and free energy, the DNA droplet shows temporary non-equilibrium dynamics, the spontaneous division here. So the next, we designed another dynamical system with the photo control of the DNA droplets. So this is a photo control of the DNA stick end hybridization is realized by the azobenzene molecules attached at the stick end part here. So when the UV light is radiated to the system, this azobenzene turns to the cis form like this. And the stick end, the hybridization is destabilized. On the other hand, the visible light is radiated to the azobenzene, turn back to the transform, and the sticky end is stabilized. Therefore, by UV irradiation, the DNA hydrogel here is changed to the liquid or the dispersed phase from the zero state. And on the other hand, by the visible light irradiation, the dispersed or the liquid phase changed to the zero state after the irradiation of the visible light. So this is the result. When the UV light is irradiated, the DNA gel changes to the liquid like state like this. And finally, DNA gels are dispersed and disappear like this. And then after the visible light irradiation to the system, the DNA gel or DNA droplet spontaneously form like this. So we succeeded in the control of the DNA phase transition by the UV light UV or visible light irradiations like this. And also the next we change the benzene insertion position and numbers so that we could control the stability of the DNA hydrogel like this. This is a dissolve the DNA, the liquid, the dissolving temperature here. So by changing the insertion position or the number of the benzene, sorry, I didn't explain the X part is the benzene here. So if we change the benzene part position and the numbers, the stability changes like this. So it means that we can control the stability or responsibility to the light can be programmed based on the benzene, the insertion position and the numbers. So based on this photo controllable system, we created the mechanical action of the DNA droplet by the controllable by the photoirradiation. So this part is a non-photoresponsible DNA hydrogel. So I mean the usual DNA hydrogel system or DNA droplet system. And the right part, the green part, so that is the photocontrollable part with the benzene at the stick end. So these two DNA gel or DNA droplet are connected with a 6-branch linker, as I explained. So by the photoirradiation, only the right part can be responsible, and then the system will change. So this is one of the results. When we introduce benzene at this point, this position, the stability is not so destabilized actually. So the original state is a DNA hydrogel state, and by the irradiation of the UV, the green DNA hydrogel turn to the liquid phase. So then the non-photoresponsible DNA particles here, the blue one, can be gathered by the DNA, the liquid green part like this. This is the result. So as you see, the white dot is the blue part, and maybe you cannot see the green part here. But by the UV radiation, the blue particles gathered into the center of this movie like this. In other case, we put the benzene these parts. So the original state of the green one is a liquid phase, and the blue is a DNA hydrogel. So by the UV radiation, this DNA liquid turn to the dispersed phase. So it means that by the UV radiation, the DNA DNA liquid dispersion makes the spreading of these DNA blue particles like this. So maybe you can see that this is the original state, and after the UV radiation, it disperses and spread. And after the visible light radiation, these liquid phase, the dispersed phase turn to the liquid phase again. So it can gather the DNA particles like this. So that we can control the splitting and the fetching of the DNA microparticles using the UV radiation to the photoresponsible DNA liquids like this. So today I don't explain so much in the detail, but the DNA active motion like that can be controlled by the benzene position and the temperature of the system like this. Okay, so we move on to the next part, the intelligent aspect of the DNA droplets for the actual cell constructions. So I mean, we want to construct the information processing ability of the DNA droplet for the construction of intelligent actual cells. So next, we considered another linker designs. So like this. So this linker has two single-stranded part here and here. We call these part are the receptor parts. So by the addition of the DNA nucleic acid, single-stranded nucleic acid into the system, that the nucleic acid can bind to this part and this part if the sequence is complementary. So then by the strand displacement reactions, these input DNA or RNA sequence can enter into the center of the linker like this. Then finally, it goes to the center, and finally, it cleaved. This linker was cleaved by the addition of these input nucleic acid sequence. So this is some kind of the sensor activity. So this is the result. If the two DNA sequences are introduced into the solution by the addition of these two inputs, the DNA droplets are divided into the two droplets like this. Sorry, it's my mistake. So in this experiment, we used the microRNA for the input. The two microRNAs are introduced into the solution. We found that the DNA droplets are divided into two DNA droplets like this. This is the result. But if the one of the input is not introduced like this, we didn't observe the droplet division like this. So this system can be considered as some kind of a logic gate of the two microRNA inputs. So we can consider this computation system in the energetical viewpoint again. So before the addition of the input, this linker is stable. So this linker cannot change to the separated states. But the addition of the two inputs, this kind of the energy landscape is changed from this gray one to black one. So the linker can be divided into the two portions of the 6-branched linkers through the intermediate, the strand displacement phase. After that, this DNA droplets phase separated by the phase separation of the two polymers, and then we have the two divided droplets like this. So in this case, the two input is not only just an input, but it can be used as some kind of the fuel. Okay, so we applied the logic gate into the more complex logic circuit, accepting 4 inputs here. The 3 inputs, if these 3 inputs exist and the fourth input is not existing in the solution, these DNA droplets are divided into the 3 parts of the DNA droplets by sequence design of the 2 types of the linkers. So the first 2 inputs cleave this one, and if the one of this input is introduced, and the second one is not introduced, this linker is divided. So this is the result. So this is the enlarged view. If the input is 1, 1, 1, 0, we have the 3 divided DNA droplet like this. So it means that we can successfully construct the more complex logic circuit using the DNA droplet systems. So finally, we move on to the third part of the construction of the DNA droplets. So the last part is cell size confinement of the DNA droplet or the interaction with membrane. So previously, the Professor Yoshikawa's group found that the genome size DNA can be specifically distributed into the phase separated Dixieland peg the phase. I mean this genome side DNA are introduced into the Dixieland droplet in the peg phase specifically. So we apply this phenomena into our system. So we use similarly the peg and Dixieland ATPase system. And we introduced two types of the DNA. The first one is sticky endless Y motif. It doesn't make the DNA gel or DNA droplet actually. So in this case, this DNA dispersed homogeneously in the peg phase and Dixieland phase, Dixieland phase too. But if we make the DNA hydrogel or DNA droplet, these large particles can specifically introduced into dispersed into the Dixieland phase only. So that we can condense the DNA system into the into the Dixieland phase in the peg. Okay, so we apply this phenomena into the microchannels. So that this is the cross-section view of the microchannel. So there is a cylindrical microwave array here. The first, the Y motif DNA with the sticky end are dispersed in the Dixieland phase. The red colored the solution is a Dixieland phase with DNA Y motif. Okay, so that is introduced into the microchannel like this. After that, we introduce the peg phase. So then in this condition, the peg and the Dixieland can phase separated with each other. So the Dixieland phase are confined in the microchannel like this. So then the DNA micro gels, micro droplets are formed only the Dixieland phase like this. So by the 2 or 3 times annealing, the DNA droplet formed to the homogeneous size DNA droplet is formed in the Dixieland phase like this. So this is very interesting phenomena because so this is the result. So you can see the DNA droplet inside the microwave. This is a top view of the microchannel. So in this case, we had homogeneous sized the DNA droplet in each wells. But if we did a similar experiment with the Dixieland impeg droplet in the bulk phase, not in the microchannels, we see that this kind of the not so homogeneous DNA hydrogels inside the Dixieland phase. So in this case, the important point is that the heating from the bottom, and it can make the convectional flow inside the microchannel, and then the DNA droplet collide at the center of the microchannel and fuse with each other at this part. In addition to the convectional flow, the second important point is the phase separation system. So because the DNA particles are expelled from the peg phase and also the Dixieland phase. So the DNA droplet was placed at the interface between the peg phase and the Dixieland phase. So it can easily collide with each other at this point. So this system is some kind of the temperature gradient non-equilibrium system. So I think this is very interesting because the non-equilibrium situation can grow the soft matter droplets. So it might be related to some kind of the origin of life story. So also by the effect of the collision that we can create the homogeneous DNA droplet. It is very useful for the application of the DNA droplets actually. So this is just a brief introduction of the formation of the phase-separated patterned DNA hydrogel capsules produced in liposome. So the liposome has a cationic lipids at the interface. So the first we produced the DNA hydrogel inside the water in micro droplet with the cationic lipids at the interface. So the DNA has a negative charge, so the DNA can gelate it at the interface of this water in micro droplet because of the electrostatic interaction between the cationic the lipids and the DNAs. So after the formation of the lipid vesicles from this water micro droplet, we had this kind of the structure, liposome with a DNA hydrogel inside. So this is the result. So this is the liposome, the edge, and this is a cross section of these structures. And then after in this case, the DNA, the phase separated gels are connected with a cross-linking the X-shaped molecules like this. So after the removal of the liposome with surfactant, we successfully extracted the inner DNA hydrogel capsule from the inside to the outside solution like this. So this is a micro scale capsules constructed by only DNA hydrogels. And also the interesting point is the phase separated the DNA hydrogel state is also remained in this case. So like this, we can control the phase separated the DNA hydrogel capsules actually, and also it might be related to the raft structure of the lipid membrane. So that maybe we can control the position of the biomolecules on the surface of the DNA microcapsule using this kind of the lateral pattern formation DNA hydrogels actually. Okay, so this is also this is a summary of today's talk. So now that we are trying to construct a dynamical DNA droplet-based artificial cells. Previously, we constructed the spontaneous division of the DNA droplets and the computational system, and also the mechanical motion of the DNA droplets. And finally, we found that this kind of the membrane-like system using DNA hydrogel or DNA droplets. So in the future, we like to combine these functions into the DNA droplet system, and we like to construct more sophisticated, more complex DNA-based artificial cells in the future. Finally, I'd like to acknowledge the many collaborators, especially my laboratory members and other collaborators in other universities, and also the sponsors. Thank you so much today.
Thank you so much. That was a really great talk. There is several questions in chat.
Yes.
If you could read the question out loud, that's going to help people watching the recording. Yeah.
Okay, so now I like to read the thank you for the fascinating to the performance of the logic gate depends on the magnesium concentration affecting the hybridization efficiency of DNA. Can you control it, increase or decrease the fidelity, changing the divergent cationic concentration. Yes, I think I think the cationic ion can change the hybridization efficiency so that we can control the fidelity of the hybridization. But if we change the cationic ions, cations, so it may affect the stability of the DNA droplet too. So I think it's a bit difficult to control both of the hybridization and formation of DNA droplet, maybe. But we can control it by investigating the concentration. And also the second question, could you do similar logic gate operating with RNA instead of the DNA? Yes. So actually, we are now trying to make the RNA-based droplets with the logic gate. So now we succeeded in such experiments, and I think we can show it later. Not so long time, maybe it will be so. Thank you so much for the questions. The next, I have a question more focused on the artificial cells role. Moving. Yes. Artificial cells role in the origin of life and the compartmentalization part. Do DNA-based droplet offer a solution to the compartmentalization problem, and what their potential role could be in the origin of life? Yes, so I think the DNA droplet can be used for the compartmentalization of the molecules because so today I didn't explain the result, but we can introduce the other molecules or the particles inside into the DNA droplet by the hybridization between such a molecule with the DNA droplets, so that we can condense some kind of functional molecule inside the DNA droplet, and I think it can enhance the reactions inside the DNA droplet. I think in such case we may be able to solve such a compartmentalization and the condensation problem of the in terms of the origin of life. But I think that the DNA droplet is not the only solution, so that it's one of the technologies to access such a problem, maybe. So the next question, so what is a temperature gradient in your droplet formation flow setup? So my answer is that here we put the microchannel device on the PCR. I'm not sure the name. The PCR device, the PCR machines. It will be flat plate, the PCR system, and so that we heated only the bottom of the microchannel. So but the top of the microchannel is not heated in this case, so that we had temperature gradient between them. So the difference between the two parts is in maximum about 60 degrees Celsius or something, but if it is cooled down, only the several temperature or something maybe. So based on such a temperature gradient, the convectional flow occurs. So this kind of flow is produced inside the microchannel, microwell. Sorry. Thank you for the questions. So the next one is it possible that these activities, if coupled, can overturn the theory of RNA world hypothesis? So the maybe the so if we produce the droplet with only the RNA motifs, that we can create such a situation. Probably, I think RNA world hypothesis can be accessed using this kind of artificial system. Maybe. Thank you for the question. And the next, and also in case of the division of the DNA droplet, if the droplets are present in substrate-rich media, in case of the division, if the droplets are present in a substrate-rich media, will it still continue to divide? So I'm not sure the question, but so after the division of the DNA droplet, in this case, the division finished because the cross-linker 6-branch linker is completely divided into 2 portions. After that, the green and blue droplets are stable, even if the enzyme and the is it okay? So I'm not sure, but maybe it will stop the division, maybe.
Actually, hello.
Hello?
Am I audible?
Sorry?
Yeah, we can hear you.
Yeah, so is it like only one-time division sort of a system, or like will there be continued division based on the size of the droplet itself?
So you mean that so if we add the cross-linking the molecule again, so maybe the 2 droplets will fuse with each other later, and after the cleavage, maybe the division occurs again. So like that. But in our system, in an experiment previously, we didn't do that. My answer is okay.
Yeah, fine.
Thank you. So the next question is it possible to perform the endocytosis using DNA droplets made of white tiles and liposome made of the DOTAP? It would be interesting to see difference between the wetting of outer wall and the liposome, the inner wall. Spreading. Thank you so much for your comments and questions. I think it will be possible, but I'm not sure it happens actually. But I think it will be possible based by controlling the wetting or the electrostatic interaction between the DNA droplets and liposome surface maybe. Thank you so much. So the next one is division of the DNA particle dependent on the size of the droplets. It doesn't depend on the size of the droplet in this case. Even if it is very small, the DNA droplet division occurs. So the final question is that for the first part of your talk, do you think the cation concentration matters for the phase separation? How you think the ionic strength affects this DNA-based phase separation? I think the phase separation depends on the cation concentration probably, because if we change the cation concentration, the fluidity of the DNA droplet will change. So I think the dynamics of the DNA droplet will change by changing the cation concentration probably. So the next question is another question I want to ask is for your DNA droplet, did you do any 4 droplet flap to confirm the exchange between the dilute phase and the droplets? We did. Sorry, today I didn't show the flap experiment to confirm the fluidity and the generated states, but we did the flap experiments to confirm the fluidity of the DNA droplet actually. Thank you so much for that question. Okay, maybe that's all.
I think you went through all of the questions. Thank you so much. Yeah, I work in logic gates myself, and I was very curious to hear your answer to the magnesium concentration question, because we deal with those things too. Thank you very much. Thanks again for giving a talk.
Yeah.
And thanks for answering all the questions.
Thank you so much.
Thank you, everyone, and have a great rest of your week.
Thank you so much.
Bye, everyone.
See you.